Hi, Robert. I like your headphones. They look very professional.
Found it's a lot easier this way.
It is, you're right. I'm Uma. We haven't met, but I've read your work. And
oh, thank you. Nice to meet you.
Nice to meet you.
Hi, go ahead.
Yeah. No, go ahead.
Go ahead. No, I just wanted to say hi, and like I heard
good
that
to see you again.
Vivek was saying that, yeah, Vivek was saying you've kind of tested everything out and looks good. Okay.
Yep, seems to be working.
Thanks to Zoom.
Yeah.
Yeah. So
better than Skype and stuff, right? It's so much, no lag.
Right, but then we don't know how much of our data is going where.
So Robin, you and Nandini are sitting in different rooms.
Yeah.
Yeah, that's right. Yeah.
Are we live already? Sorry.
No,
Yeah.
we are live, but not on Facebook, but the attendees can see us.
Okay.
Yeah.
Hey guys, our audience mostly would be from India, or like what your experience over the last few days?
A lot of Indian students, right?
I see.
Okay, so we're going live now.
Vivek?
Yeah. Good evening and welcome to the last session of this year's Humboldt Day India events. These events are being jointly organized by Dr. V.V. Robin from IISER Tirupati and Professor Ramakrishnan and I, Vivek from the National Center for Biological Sciences. These events are supported by the Museum and Feed Station Facility, NCBS, and the Biodiversity Collective, Collaborative. As moderators of today's session, we have Dr. Nandini Rajamani, Assistant Professor, Indian Institute of Scientific Education and Research, Tirupati. Nandini's Sciurid lab studies the behavioral ecology and evolutionary biology of animal populations, often within the context of environmental and habitat change. They work across the multiple landscapes in India, including Trans-Himalayas, Western Ghats, and Eastern Ghats. Over to you, Nandini.
Hi, Vivek. Thank you. I'm excited to be here, and this seems to have been a great meeting for the past four days or 5 days. We've had exciting talks and a lot of interaction. I'm happy to introduce the speakers of the day. We have 3 talks today. We have Dr. Robert Moyle, who's going to start, followed by Dr. Sampath Seneviratne and Dr. Martin Packard. So, yeah. So we'll start with Dr. Moyle. We'll have questions after each talk, so we'll have a little bit of a break between the speakers, maybe about 5 minutes, and then we move on to the next. We will also have space at the end of the session for general questions, in case people have questions and we can't answer them right after someone's talk. Dr. Robert Moyle works on the historical biogeography of pantropical groups, patterns of endemism, and drivers of diversification in Southeast Asia, as well as the tropical Pacific. And he also works on the life history traits, evolution of life history traits within tropical groups. He's a professor and curator at the University of Kansas in the US. And over to you.
Thank you very much for that introduction. Let me go ahead and share my screen. Okay, hopefully you can all see that screen now.
Yeah.
All right, thank you very much for the introduction, and thank you to the organizers for inviting me to give this talk today. My only regret is that we can't actually do this in person, but I guess Zoom is good enough. Considering the occasion for this talk, which is a celebration of the field of biogeography, named after one of its founding inspirations, I've decided to revisit a classic pattern that's attracted attention from biogeographers for, gosh, more than a century, 2 centuries at least. And this pattern is disjunct distributions. And when I say disjunct distributions, I mean great isolation, not just separation by a river, but generally great expanses of ocean. So one example of this, of course, are isolated oceanic islands. And you've heard a talk earlier in this week by Dr. Gillespie about this, and fascinating faunas that evolve on these isolated archipelagos. But another example are continental disjunctions. And so here are a few of many, many examples out there of widely separated groups that are obviously closely related, but broadly separated by wide expanses of open ocean. This is a classic question in biogeography because it's been debated for such a long time, but also because it has great lessons in sort of how science progresses. Sort of how we learn how paradigm shifts happen when a new idea comes along and sort of everything changes. It also shows how we need to be multidisciplinary in our work, but often aren't, unfortunately. And so what I'd like to talk about today is just a very brief sort of history of some of the main paradigm shifts that have happened in the biogeographic realm with regards to widely disjunct distributions. And then I'll use a couple of sort of recent examples from my own work of advances that we've made in Passeriformes. These are the perching birds. And the advance there is more about the timing of diversification, not so much pattern. And then the Carassiformes and Trogoniformes, which it's a very similar pattern, so I'm not sure if I'll have time to talk about both. But the interesting item there is the extant distribution versus this fossil distribution. And I'll talk about that a little bit. And common themes are going to be sort of the advances that have been made in phylogeny reconstruction, estimating timing of diversification, and definitely how to incorporate fossil data into our molecular data from extant taxa. So if we go way back in history, initial attempts to explain these disjunct distributions really fell into 2 classes, 2 major paradigms. You had the dispersalists, who felt that animals dispersed out from a center of origin or ancestral area and dispersed long distances to colonize these new areas. These were people such as Darwin, and Wallace, not in the beginning, but later came around to also be a dispersalist. In contrast, you had the extensionists, and they felt that this dispersal across vast oceans was unlikely, and so they felt that land bridges more adequately explained how these taxa got to the different continents. These were called the extensionists. These are people such as Hooker and Lyell. As we learn more about ocean basins and the great depth of many of them, the extensionist paradigm sort of fell out of favor, leaving the dispersalists for a while. Now, all of this changed again when we learned about continental drift. I should mention, you can see in the image here, if I can pull up a laser pointer. So if you look at the Atlantic basin, I believe it was Alexander von Humboldt who first noticed the incredible fit between the coastlines, for example, between South America and Africa. Of course, he didn't know then what exactly that meant, and it wasn't until Alfred Wegener in the early 1900s who actually formalized the idea of continental drift, that the continents actually moved. Of course, this wasn't accepted either because that seemed like a crazy idea. Through time, though, with ideals like paleomagnetism of the seafloor, around the 1960s and 70s, this became widely accepted. And suddenly, there was definitely a paradigm shift in biogeography because now you had another explanation for these disjunct distributions across the globe. Biogeographers sort of en masse started to look at this as a possible explanation. And one of the patterns, or one of the hypotheses that came out of this, was the Gondwanan hypothesis. So the southern continents, South America, Africa, India, Madagascar, Australia, New Zealand, and Antarctica, were all a supercontinent Gondwana, and they drifted into their current positions, and this could explain these distributions. So I only picked a few out, but this became sort of the latest rage, explaining these disjunct distributions via Gondwanan vicariance. You didn't need to invoke this long distance dispersal anymore. So these are a few of the many, many papers that came out about it. Now, one group that this seems to fit pretty well and has been applied to are the passerine birds, the so-called perching birds, Passeriformes. So diversity here, phylogenetic relationships were somewhat uncertain until DNA sequencing, but the early days of DNA sequencing, about the turn of the century, it became clear that the relationships were as such, with the first branch in the group being the New Zealand wrens. Only a few species, don't fly very well, entirely isolated on New Zealand. You have the suboceans here, two clades, one almost entirely restricted to the New World tropics, the other to the Old World tropics. And then finally, this massive clade up here, the songbirds, the Oscenes, which in contrast to the other groups have diversified across the entire globe, colonizing all continents and diversifying into almost 5,000 species. And that's sort of the part of the tree that I'll focus on here today. Now you can see from the continents involved, sorry, I should say songbirds, it's pretty well established that they arose in Australia. So Australia, South America, New Zealand, these are all Gondwanan landmasses. It certainly fit the Gondwanan pattern. So in the same year, Erickson et al. And Barker et al. Both published papers that seized on this idea that you could explain passerine diversification via Gondwanan vicariance. This is a figure from Per Erickson's paper. And this is an interesting hypothesis, because with it, you not only explain pattern, but you can also get at the time frame of diversification, because there are pretty good date estimates for when the continents split apart. One of the best being when New Zealand split off of Gondwana at about 82 to 85 million years ago. So this date was used. Now this is a hypothesis, and unfortunately hypotheses are out there to be tested. But in the bird world, and I was guilty of this as well, people didn't really test it, they just used this date to date all sort of diversification within the passerines. And so there were dozens, maybe 100 papers that used this date, either directly or indirectly, to infer sort of what happened in the passerines, which is really exciting that we could nail down these dates. One problem, though, was what it actually sort of caused to be inferred in parts of the tree. So the songbirds, what I'm interested in here, they had several lineages that dispersed out of Australia into the rest of the world to diversify. With this Gondwanan vicariance scenario, and using New Zealand as a date, here's where Australia was when all those lineages dispersed out, presumably up through Asia to the rest of the world. So we've gotten over invoking dispersal by using Gondwanan vicariance, but in turn, that has caused us to invoke long distance dispersal to get out of Australia. So maybe not entirely satisfying.
Can you hear? We can't hear you anymore. We can't hear you.
We still can't hear you.
It was fine just a minute ago.
You're not muted here, so it has to be the microphone. Can't hear you.
We can't hear you.
I think you forgot, right?
Yeah. No, no. I think we lost you 2 slides ago, or 1 slide ago.
Okay, can people hear me now?
Yeah.
Okay, I
Thank you.
changed it to my camera instead of my headset. Okay, back to where I was. All right, so I was mentioning that what's needed are independent calibrations of the time frame of diversification. And what happened pretty much in the last decade were paleontologists working really hard to identify fossils in a phylogenetic context with character-based placement. Also, paleontologists working with neontologists to better incorporate fossil data into molecular phylogenetics. In the past, pretty much the neontologists, molecular systematists would get results and they'd look at the fossil results and they would just sort of compare them instead of integrating them together. So 2 of the first papers to do this in birds are these 2 papers up here on the screen right now. They looked at all of birds, sort of the higher level relationships of birds. And that was important, and that's sort of what got all the press, these genomic analyses of birds. But another thing was they did a really good job of incorporating many, many different fossil calibrations to try to date divergence times. And now you remember that the separation of New Zealand from the rest of Gondwana was about 82 to 85 million years. And that's what people have been using to calibrate ages within the passerines. So what sort of dates did they get when you use independent fossil calibrations for that same node in the tree? Well, very different dates and much more recent dates. And so if these are correct, that sort of casts passerine diversification in an entirely different light. So my lab, in conjunction with several other labs, looked more in depth at the passerine radiation to see sort of what fossil calibrations could tell us about diversification within those groups. Here are the 2 phylogenies. Too many details to talk about right now in this talk, but one thing to note is we also got a much more recent date for the splitting off of those New Zealand wrens, the Acanthocytidae, which is very much in line with those 2 papers I just mentioned. One thing that is, it doesn't make it right, but it's very satisfying, is that what this causes us to infer is that the dispersals out of Australia to the rest of the world, where they colonized the whole globe, are now inferred to have happened between 20 and 25 million years ago. Well, this is when geologists calculate that Wallacea was rapidly uplifted, not quite into a land bridge, but at least stepping stone islands. And that is when passerines dispersed out of Australia. So this is a combination of advances in DNA sequencing, fossil calibration, and also incorporating geology. And it also shows that a single paradigm can't explain passerine diversification. You have vicariance, you had dispersal, and I guess via Wallacea, you sort of have land bridge extension as well. I guess the extensionists would be pleased. Now to shift gears a little bit, talk about a similar but different situation. With the passerines, the Diocenes were globally distributed. But here's another distribution, one of the ones that I'll be talking about, and these are much more restricted to the tropics, and so even more isolated, I would say. People have taught, biogeographers have talked for decades, centuries, about how to explain these disjunct tropical distributions. The groups in question, there are many. I'll be talking about 2 of these 3, but I've worked on them, so I'll put them all up here. Trogons have that pantropical distribution, barbets and toucans, and then the broad order of the Crassiformes, which includes kingfishers, rollers, motmots, toadies, bee-eaters, a whole diversity of families broadly have that pantropical distribution as well. And this is what these distributions look like for those 3 groups. You'll note that the Crassiformes doesn't look quite like the others because it seems to extend up into the temperate regions, but the vast majority of the diversity is in the tropics. Only a couple of kingfisher species, which are mostly migratory, extend up into the temperate regions in the north. Another curious aspect of these groups is that their oldest and almost entire fossil record, if you'll see down here on the Trogoniformes map, is from Europe and North America. And this holds true for all three of these groups. In the Eocene and Oligocene, fossils of all three groups from Europe and or North America. So far, far from their current distribution. So we've worked on some of these groups. Here's an example. This is a phylogeny of the Trogons. Genomic data did a nice job of sorting out the relationships of the group, but it's difficult to talk about origins when, as you can hopefully see on the picture, you have an African clade, cursor is hard to control, there we go, an African clade, sister to a group of Asian Trogons, sister to New World. Three areas, all closely related. Hard to infer what's going on at the base of this tree down here in terms of ancestral areas. And sure enough, if you just optimize sort of ancestral areas, and that's what these figures are looking at here, sort of this basal, the stem lineage, you can't determine where it might have occurred. It was either Africa or Asia, and you can't see that pie in there, but even possibly the New World. However, if you incorporate those fossils, so in the past, people would have taken this phylogeny in reconstruction, said fossils are in Europe, and just left it at that, say there's disagreement. But now with methods to incorporate fossil calibrations for biogeography and dating, as you can see over here, these are two fossil taxa that were included in the analysis. Now the reconstruction overwhelmingly supports an origin of Trogons in somewhere in the Asian landmass. And so by incorporating fossils, we get a much different signal from the data than we might have otherwise. Almost the exact same pattern in the Crassiformes. This is a much larger phylogeny. This is actually a complete species level phylogeny of all the Crassiformes using ultra-conserved elements. The thing to note here with all those red arrows that I've put all over the phylogeny is those are fossils that have been placed phylogenetically in the phylogeny that can aid in divergence time estimation as well as biogeographic reconstruction. So this almost entirely South American, African, Southeast Asian, Australian group is actually inferred to have originated in the Palearctic with a shift to the Nearctic and eventually settling down into the tropical regions. So a very, very different result than would have been obtained if you just used extant distributions. So those are two examples. You may ask, well, okay, how widespread is this? Well, we're realizing in birds, it's incredibly common. So hummingbirds are endemic mostly to South America. A few migrate into North America. There are, I believe, Eocene and Oligocene fossils from Europe of hummingbirds. So this South American distribution is relictual and misleading in a way. Mouse birds, a small radiation in sub-Saharan Africa, have a deep, deep history, once again, in North America and the European fossil record. Frogmouths, restricted to the Australian region, extending up into South and Southeast Asia, have fossil record in North America and Europe. And finally, one of the most bizarre ones, the Hoatzin, this bizarre looking bird here, endemic to South America, no close living relatives, actually has fossil taxa have been found representative of this family from Europe and Africa. So once much more widely distributed. And so it certainly shows that just judging biogeographic history based on extant distributions can be extremely misleading. Yet that is what we do a lot of the time as neontologists in looking at biogeographic history. So in summary from these two, these disjunct distributions have really vexed biogeographers for decades, if not centuries. But recent advances in understanding Earth history, more collaboration with paleontologists and paleontologists integrating the fossil record into these studies, DNA sequencing to use genomic data, and methodology, just the theoreticians and developing methods to accommodate all these different types of data have made great advances. And it also goes to show that, you know, the paradigm swings back and forth, dispersal, vicariance, extension, it's old, it's young, back and forth. And usually it's probably a combination or somewhere in between. You know, you might ask, so is the pendulum still swinging, or you know, have we gotten it? I would say it definitely is. You know, new fossils, there aren't many fossil strata from certain continents. And so uncovering more of the deep biogeographic history of these groups could really change everything that I've just talked about in a way. And so I think there's still a lot of advances to be made in any of these broadly distributed groups. With that, I'd like to thank funding agencies, many countries for permits for doing field work, a ton of collaborators on all these different papers, too many to list. And then of course, tissue loans from various institutions who are out there doing the hard work of collecting specimens and tissue samples so that we can do this kind of work. Thank you very much. And I'd be happy to take any questions. Looks like we do have a few minutes.
Thank you for that fascinating talk. There was so much covered in that. It was amazing. Can we have questions? Can people type their questions into the chat box? We have one.
Now, unfortunately, I can't hear.
I can read it out if that's easy. It's in the Q&A box. What is the correct biogeographical distribution pattern for indigenous birds?
Well, Indian is, I think, what.
Abhishek, did you mean Indian birds?
Yeah, let's.
Okay, so the next question, could there be a spatial bias in fossil research? Is there more research from the Northern Hemisphere? I think Robert can't hear us. Robert, can you hear us?
Just barely. I don't know why
Okay.
my headphones seems to have conked out.
All right, just one question, and if you can hear it, then we can take it now, or we can take it after the talks. Archita asks if there could be a spatial bias in fossil research. Is there more from the Northern Hemisphere?
Actually, let me just go into here. So I can read the comments.
In the Q&A section, the Q&A.
I can barely hear. I'm so sorry. I can't believe this has happened.
We can
Well,
just
come
come
back.
back later, Nandini. We can probably do these later.
Sure.
Or, yeah.
We
Because
can
there's
come
another
back.
question, I think,
So,
on
I'll just do my, is it okay if I just go ahead and answer some that I see posted up there?
Yes, please.
Okay, so I see one right here. Could there be a spatial bias in fossil research, more research from the Northern Hemisphere? Absolutely. It's a combination of bias in activity and attention, but it's also just some of the vagaries of where there happen to be really good fine-grained fossil strata that happen to be exposed. So it's a combination of the two, but that's definitely a problem. Yeah, we're finding lots of fossils in Europe, but that doesn't mean that that's the only place that we expect them to be. Absolutely.
the next one is also, the next two are also, you can see them. Toucans. Oh, that's one.
I'm sorry, I can hear people talking. It's my tiny little speaker under the monitor that I can hear a whispering.
There are two more questions.
Two more questions. Okay, I can go to the bottom. Do you think the warm climate and widespread tropical habitats around 25 million years ago helped passenger diversification? Yes. This is something that I probably should have mentioned in it. So, you know, the mystery, you know, so why are these differences in distribution happening? So this happens in both parts of the talk, the passerines as well as the trogons and crassiforms. In the past, especially somewhat in the Oligocene, late Oligocene, but then also in the early Eocene, the Earth's climate was much different, much warmer. And so you had tropical and subtropical habitats extending far further into the higher latitudes than they are today. So in the early Eocene, you had crocodilians, flying lemurs up all the way to the Arctic Circle. And so that explains why these seemingly tropical groups could have this incredibly broad latitudinal distribution. And what's happened is not that they've adapted to suddenly become tropical, it's that the tropics have receded into what we now know as the tropical regions. And so that is probably the most likely interpretation, exactly. Toucans in America and the hornbills of Asia, Africa. Is this a result of convergent or divergent evolution? You know, I haven't looked at those specifically, but it would seem to be fairly convergent. I mean, they're not that close. They're not closely related enough that massive bill structure would be a synapomorph by any means. And so it seems like, at least in general morphology, that would be convergent evolution. Now, as far as function, that's difficult to say. We see a bill and we usually think feeding. It's got to be feeding. But there's other uses for bills, from parasite removal, possible to thermoregulation, and signaling too. A lot of toucans have ornately patterned bills, and some of that might go into sexual selection and things like that. So there's a lot of different aspects to look at there, and I'm not sure how all those aspects actually correspond between those two groups, but good question.
All right, shall we move on, and we'll save the other questions for later?
I'm sorry, should I take one more? Okay.
We can move on.
Later.
I could pretty quickly. Do you face impediments while sourcing genetic material from different countries? Yes, it's very difficult. And in a way, it should be. You know, countries have domain over their natural resources. They determine sort of how things are going to be used or not used. Hopefully through more collaborations with researchers in different countries, you know, this can become easier. But definitely getting source material is a constant sort of limitation on the work. But as you can see, we're still making advances. So it's not stopping us by any means.
All right. All right. Thank you so much.
Thank you.
And we hope you'll stay around for the end in case there are other questions. So we move on to our second speaker, Dr. Sampath Seneviratne, who's going to talk to us about some of his fascinating work in the Sri Lankan island. So Sampath is based at the University of Colombo in Sri Lanka, and he heads the avian evolution node, where he uses birds as model organisms in the study of island biogeography, causes of endemicity, and the evolution of novel traits. So today he's going to talk to us about biogeography in Sri Lanka, from Wallace to warblers.
Okay, thank you, Nandini, and thank you everybody, the Indian team, to invite me for this. We had a small event at Sri Lanka in the Sri Lankan Herbarium a few days ago, celebrating Humboldt and his legacy. So this is a kind of extension, and while I'm getting my slides up, so it's been a very interesting week, very interesting talks, and okay, I think I'm good, and then I'll get my laser pointer. Yeah, and so it's been great actually. I learned a lot, and also Humboldt is, I think, truly. You can hear me okay, right? Yeah, okay, cool. He's really a forgotten hero in our specific areas of study, and also somebody who put conservation biogeography in one plane. It's in that era. It's truly amazing. So here what I try to do, I've tried to give a kind of a snapshot in the south of the peninsula point of view, to kind of give you an island biogeographic standpoint. I call it warblers, Wallace to warblers, partly Wallace, like my personal hero, and an Asian man, kind of belongs to Asia, somebody from middle class, and you know, so it represents a lot us than some of the great evolutionary biologists and biogeographers of the past. So anyway, so let's talk about, I will start with Wallace and end with warblers. Just to give a quick snapshot, I think I will have enough time for my 30 slides. It's a kind of a visual representation of what I try to say. This is Ptolemy's depiction of the world. As you can see, this is Taprabanika, which is Sri Lanka. This is Europe, UK, Africa. The southern borders were not clear because at that time there wasn't clear, actually known to the Western world. He's coming from Alexandria, so it's somewhere in Europe. So Sri Lanka is big, and India, I think this is India. Probably he never been to India and didn't know the existence of it, or the southern part of India probably, or the shape of it. Whatever it is, he knew the Indian Ocean. Sri Lanka, kind of sizable portion of his knowledge, at least the way it looks. Partly the reason is the cinnamon, the black pepper, and cardamom. One of the prized spices that went from Western Ghats and Sri Lanka to Europe. So the botany, or truly speaking, essence of botany, were there in Europe, in the Western, the modern Western science. But it was basically dominated by Moorish traders. But with the opening of the southern routes, the Western powers try to kind of reach or approach the East, in this case, the South Asian region, for this wealth. So the biogeographic standpoint, there's a meaning because these are endemic herbs, endemic spices to the region for the southern Asia and Sri Lanka. And also it's represent that specific alkaloids and all, which is also a kind of a biogeographic element. And just to get back to the legacy of Humboldt, a great scientist, a naturalist, and as I said, put ecology, global change science, I think, in the animal patterns, especially in the South American mountains, and the study of some of the European mountain ranges. Truly amazing. So that with that, let's get back to the to South Asia, and this Western knowledge that I think catalyzed by Humboldt's work and some of the early work, actually reached to our parts through these steamers, and in the process of them following the aromas, rich aromas of spices and ivory and so on. So with that, in Sri Lankan standpoint, the Ceylon, Taprobania, Ceylon, all these are different colonial names. Heladiva for the Sinhala vernacular name. So Sri Lankan biogeography begins in my opinion, with Henry Tryman, a botanist who described flora of Ceylon, Sri Lanka, and there are many descriptions of flora in the light of its economic value, especially the value of its spices, its medicinal and culinary values and aesthetic values. But Tryman is the first to kind of put the biogeography in place by comparing Ceylonese angiosperms with the Indian forms and with the Southeast Asian forms. So the pattern or the tradition begins there for the island, and then the father of biogeography, the guy who lived in Asia, a good part of his quality academic life was spent in Eastern Asia, had many visits to Sri Lanka on the way from England to Eastern Asia, because Point de Gaulle, the Port de Gaulle, the Gaulle port being one of the stopover sites. And also he had this special friendship with Anand Kumaraswamy, an academic that he met in England, somebody with the Indian affinity, lived in Sri Lanka, Sri Lankan, and in this context, Wallace had collaborations not about science, but about the land rights, especially the unjust and acquisition of land from Indian and Sri Lankan farmers by the colonial power. So he was communicating with Ananda Kumaraswamy about the Swadeshi movement that they had in the continent. But very interestingly, and going into his domain, he commented that the Ceylon, more closely connected to India than Britain is to Europe. Many animals and plants are different from those found in India and peculiar to the island. So the somebody who's very good at, very versed at the spatial distribution of plants and animals, he picked this very early in his the history of Sri Lankan biogeography. Lawton, the Lawton sunbird named after one of the Dutch governors, and then the Linné himself used some of the material came from Lawton in his early classifications. And in the light of birds, where I tried not to use birds along this half an hour, but eventually dragged back to it, and I thought, okay, I will let the birds dominate later in my slides. But in my bird world, Vincent Legg is the hero, where about 300 plus species were actually described or documented from Sri Lanka during his 8 years of his time in 1872 to 1880, and published this 3-volume large series, the history of the birds of Ceylon, considered one of the best books written in his era on in the British Empire on natural history, with some amazing illustrations. And then the Herdman done some work in Mannar Basin on invertebrates. So that's eventually led to the beginning of Sri Lankan tradition of biogeography with the great P.P. Darniyagala, the Sri Lankan, the native biogeographer. He is a prolific writer. In his 39th year, he became the director of the National Museum and published about over 379 or 380 books and papers, including items on ichthyology, herpetology, ornithology, mammalogy, paleontology, and prehistory. A truly Sri Lanka's version of Wallace. And he talked about human evolution, the use of tools by the Adivasis, the Aborigines of Sri Lanka, the reptiles, fish, and this small piece about Darniyagala with his description of Mesoplodon hotaulla. This is a beaked whale described by Darniyagala in 1960s, but he named this as hotaulla, which is the hota means the beak, ulla means the pointy beak whale, so named after Darniyagala's beaked whale. But soon it was synonymized with the Mesoplodon ginkgodens in Japan after 2 years of its initial description. But recently, some of the cetacean biologists from New Zealand came across some strange dead whale, like whale meat, and they've done some DNA work and realized that that from Palmyra atoll in the southern Pacific and the tropical Pacific, and then realized that it's not matching with the specimens of Mesoplodon ginkgodens. So they eventually followed the trail of Darniyagala's description and came to Sri Lanka and done some sequencing and showed that Mesoplodon hotaulla is actually a true species. It's a tropical version of the ginkgodens. Anyway, so Darren Eagle had that shrewd, sharp mind throughout his career. Anyway, that's a kind of very quick snapshot. Let's get back to the thrust of my topic. The island of Sri Lanka. It has a distinctive central hills about the height of Palani Hills of Nilgiris and Ootis and all, about 2,500 meters. It has a distinct wet zone in the southwest and a seasonal dry plateau in the plains in the north and northeast. And a central highlands where fairly high rainfall. So this makes the island like 1/4 of it about wet zone, and the rest is kind of seasonal dry zone. As many speakers had mentioned over the past few days, the beauty of Indian plate is that it has this the biotic ferry effect, where it moved from about 49 latitudes south in the southern hemisphere to its present location in the northern hemisphere. Sri Lanka moved about 50, slightly more than 50 degrees towards northwards across the Indian Ocean over the span of about 100 million years, and it's now located 6 degrees to 10 degrees north of the equator. So this island is nestled in the northern Indian Ocean, and it's quite green compared to the southern India, and it's in the mountains especially, it's very green cloud forest with very high endemicity and all. So let's see what are its characteristics. This is the southern wet zone, lowland wet zone, which is dominated by the tropical rainforest, and the tropical rainforest of Sri Lanka is dominated by the main trees of it consisted of Dipterocarpaceae. So the lineages coming from the Gondwanan past. I'll kind of summarize, or I'll kind of sketch few main points of Sri Lanka's biogeography. One main aspect is that it's quite unique, a biogeographic zone from its Indian, the rest of its Indian plate in the southern Indian Western Ghats. It's been shown by several authors. One of the pioneers and the key papers is this Boswell et al., which shows that of the sedentary forms like crabs, fish, and then the reptiles and amphibians, that it is quite unique. It has its own identity, it has its own endemism, and it has its own radiations. And that kind of resonates what Wallace had been telling about 2 centuries ago a century ago on its differences from the Indian mainland. So even though Western Ghats and Sri Lankan wet zone is one continuous kind of a geological formation, it has Sri Lankan side has unique features. So that is itself one unique feature. One of the unique features, being an island, it's a continental island, is that it has high endemicity. In from spiders to mammals, these are some of the data that we have, like groups that we have a good representation of data. You can see that the red means the number of endemics, and then the green means the total number of resident species. Even groups like vegile groups like birds and mammals, and the sedentary forms like spiders, snails, and all, there is a relatively high endemicity within the island. And one of the classic cases is the freshwater crabs, where 50 of the 51 species are endemic to the island. And similarly, birds, which is of the bunch, the most fragile, I think, the ability to fly. And the passage between Sri Lanka and India is just about 25 kilometers. And that is also dotted by kind of series of stepping stones. So the birds theoretically would not find that difficult to cross. They are showing very high endemicity. This is the percentage out of 508 total ever recorded in Sri Lanka. 508 so 34 endemics. But in the resident forms, of when you consider the subspecies variations, about 1/3 of all phenotypic variations of Sri Lankan birds are unique to the island one way or another. So even then have fairly high endemicity. And Sri Lanka shows rapid radiations. Classic like what the Indian biogeographers has been showing over the past 2 decades in the Western Ghats, where these rapid radiations that you see in reptiles and other groups in Western Ghats is represented in the Sri Lankan side as well, with freshwater fish, with the skinks and geckos, and in this case, the pseudophylatus tree frogs. The rapid radiation is quite interesting, because that shows that some probably due to the Deccan furnace effect of the climate change that kind of wiped out some of these lineages after its drift from the mainland, from the Gondwana to the Laurentian mainland in the past 50 million years, or due to some other changes in the topography or climate, would have wiped out some, and that would lead to this radiation. Either way, the Sri Lankan context of that part is still a mystery, but Sri Lanka also has the same rapid radiation that's been shown in Western Ghats. Similar to Western Ghats, Sri Lanka also has these phylogenetic relics. This is a study that Robin, Uma, and Nelum and all, some of us done a few years ago, where the Sosterops silonensis, the Sri Lankan white eye, is kind of isolated and isolated within the phylogeny of these white eyes, and white eyes has one of the highest rate of divergence for any vertebrate group. So it's an interesting group, widespread Asian passerines, but the Sri Lankan one is kind of the basal in the known white eye phylogeny. Then this has been shown in other groups as well. So the relics is another interesting thing. In Sri Lanka, this there's fairly kind of a clear pattern of climate-induced divergence, partly because it's very green. Its wet zone is insulated by dry zone of Sri Lanka and the dry zone of India, and salty, the Pork Strait. So like groups that has the ability to fly across, as I mentioned earlier, also shows this, this pattern of changes. This is the flameback woodies, where they are yellow in the mainland, and then they get red in Sri Lanka. Let's see what, like, even in the two distinctly different genera, the Chrysocolaptus, the greater flameback group, and the Dinopian, the lesser flameback group, both the members are yellow in the mainland, but in Sri Lanka they are red. So we've been working on this, looking into this in different aspects, from genomics to ecology. Saminda recently finished his PhD on this group. So we are kind of, it's kind of one of our pet models. There also you can see that it's gradually changing into the different form genetically and phenotypically as it moves to the southern part of the island, from yellow form to a series of orange ones to red. Similar pattern has been seen in drungos, where one of my students, PhD students, Sanjay Virakud, is working on, and we managed to now kind of iron out the phylogeny, and it shows that again the Sri Lankan wet zone type is distinct lineage, and again slightly different from its mainland. Something not so well established idea is that there's a fair bit of evidence of incipient speciation, the speciating groups found in Sri Lanka. And this case in the Northern Ancestry, one of my undergrads right now quite passionately working on this group, Jude, on these Kentish plovers. And what we've seen that the Kentish plover, the Sri Lankan and South Indian, South Indian form, the Siboimi, is quite different in certain cases, not so quite different in certain other cases. So we can start to see a pattern of a population being isolated, and a subspecies is formed, and a species is forming, which is from the northern lineage settling in Sri Lanka. And the final two slides, I think I'm good with time. The warblers. So we've started looking into this, the mystery bird of Sri Lankan mountains, the Ceylon warbler, which doesn't have a close phylogenetic relative. Some put it as a babbler, some place it as a bush warbler of the Far Eastern Asia, like China, Eastern China, and some placed it under its own monotypic genus. So it has been a case for the past, I think, 150 years, ever since it's been described as the Ceylon warbler. So we've done some work using fresh samples and its ecology and measurements of a whole bunch of birds. And so finally we came up with a relatively resolved phylogeny. The interesting thing is that the Ceylon warbler is actually a grass, like a close relative of a grass. But by the way, the Ceylon warbler considered as one of the classic postcard relic, because it isn't believed to have its closest relatives in the Eastern China. So it was our postcard relic. But when we done the phylogeny, it's sister with the broad-tailed grassbird and the bristled grassbird, and then the rest of the grass birds of Africa. So its African origin is fairly kind of started to appear, and so we are working on its the phylogeny and its ecology to kind of to show its affinity to Africa. And again, this African bird somehow crossed these barren, once upon a time, I think much greener central plains, and then enter into India, and then the grass birds are the reminiscence of this its relatives and its past from Africa. Right. I think that's kind of what I have to tell about to show the value that this island brought into the South Asian biogeography. There's a lot that we can study and to understand about the broader patterns of the Indian plate, its movement, its radiations, and it's a good comparative kind of like a platform for the Indian the biogeographers to study. And from us, from the south, the Western Ghats and the Eastern Ghats and the Himalayas poses whole interesting comparisons to understand more about what's happening in these rainforests in Sri Lanka. I'm the spokesperson of many other people. In a rush, I couldn't add some of the institutes and supporters and some of the students that contributed to this, but it's a collaborative work, and some of you has been helping us over the years, and hope to do more in the future. Over to you, Nandini.
Thank you, Sampath. That was a really interesting talk. Thanks for taking us through the history of Sri Lankan biogeography, and then ending with some fascinating studies about birds. There are quite a few questions. Would you like me to read them out to you? There is a Q&A box.
Yeah, please.
Okay. So Abhishek asks, what is the cause of herpetofaunal composition between the Indian Peninsula and Western Ghats biogeographic provinces? They've been increasing in their distinction and overlap in the number of species, and due to parallel increase in endemics.
Okay, yes, that's an interesting thing. I think we started to see that in not just herbs, in other groups as well. Partly, I think there are. I mean, I don't know the answer, but my guess is that, and also talking to some of the leaders in your part of the continent. That there are quite a fair bit of parallels especially in the wet zones of the Western India, South India, and the Western Sri Lanka. So there's a whole set of parallels that causes convergence. And in the isolation, like not just between India and Sri Lanka, but between the Palghat gap and then the Rajmahal gap, and then like that. So the valleys that along the Western Ghats would isolate them, and then you have a parallel climatic changes have taken place. And then on top of that, there's the habitat heterogeneity of, especially within these small like pockets, and in the differences between the western side and the eastern side. So there's parallels and parallels in differences, and then identity, unique climatic microclimates. So that creates a situation where you have divergence, convergence, divergence, convergence. So I think so that contributes to the endemicity and this radiations that you see. I think that's the question, is it?
Yeah, I think something like that. There are a couple more questions that have a few votes. How are the dry zone species in South India and Sri Lanka related to each other? Are the species fairly similar, or is there evidence of park strait acting as a barrier?
Good question. I wish I know the answer. Eventually we'll ask that 20 years from now. I think the Robin and like Roy, we all would like to see the answer for that. But the interesting thing about dry zone, the wet zone is, yeah, it is the main cooker in terms of speciation. That's where the big divergence and radiations have taken place. But dry zone is an interesting place because dry zone has, in a way, much more contrasting changes, differences from riverine forests to rain shadows to and then the valleys, dry valleys to wet valleys, and so on. So I think there's a fair bit of heterogeneity within that as well. Even though, since in the light of the very heterogeneous wet zone, I think people tend to overlook that. Especially in Sri Lankan context, Sri Lankan dry zone is not really dry because it has the permanent rivers flowing, so as in some parts of India. So there are, in more broader groups, I think there's a fair bit of similarities, at least the lineages are kind of shared. But then again, I think the radiations are there. It's started to see that pattern more and more. So yes, in a broader sense, wet zone has more diversity and more endemicity, but I think dry zone comes quite close.
All right. So in the interest of time, maybe we can have the next talk and come back to questions, because there are a lot of questions. There's a lot of interest clearly between India, Sri Lanka comparisons, but maybe we can come back at the end of the next talk. Yeah.
Great.
Thank you so much, Sampath.
You're welcome.
All right. So,
thanks.
Yeah. So we move on to our last talk of the day. Dr. Martin Packard, who's based at the Senckenberg Natural History Museum in Germany. He's worked extensively on the Tibetan Plateau, the Himalayas, and he's worked on the evolution of present biodiversity patterns using geological, climatic, and biological information. His phylogeographic work on various passerine species gives us a unique insight into the region, also referred to as the third pole. So Today he's going to talk to us about mountain diversity or biodiversity on the planet, specifically with respect to birds.
Hello to you all. Can you hear me? Yes?
Yes.
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 Okay, fine. Then hello to you all. Thanks, Nandini, for the nice introduction, and thanks to Professor Ramakrishnan, Dr. Robin, and to Vivek for organizing this event. We had a fantastic week with great talks, so it was absolutely enjoyable so far. And I'm happy and honored to give the closing talk of this event on the biodiversity of the Tibetan Plateau, and I'm now trying to share my screen with you. Does it work? Can you see the screen?
Yes, we can.
Okay, but I can't anymore. So I'm having a look at my, okay, but I can see my own screen now. Okay,
Yeah.
fine. Yeah, we're still in the Humboldt year. Actually, it started last year, celebrating Humboldt's legacy 150 years after his death. And we had many events worldwide. We had a meeting in Frankfurt. We had the Humboldt issue of the Journal of Biogeography. And many of the research reports and talks were dedicated actually to mountain biogeography. And this is a couple of slides and pictures you have already seen during the last days, so they look pretty familiar to you. And actually, his work, Humboldt's work on the Americas, on the biogeography of the Andean organisms, for example, are pretty well known. And you have already also seen the travel itinerary from his expedition to the Americas in 1799 to 1804. But what is perhaps less known is that Humboldt was also very interested in the Asian mountain systems, because Humboldt already had a quite global perspective on biogeography. He was very interested in comparison patterns across continents and also across mountain systems. And he was always eager to travel the Asian mountain systems. He also wrote a small chapter on the Himalayas and his views of nature in 1826. And 3 years later, he had the chance to travel to Asia, actually. Here you can see the travel itinerary for his expedition to Russia. So he came as far as to the Russian Altai, but unfortunately, he failed to fulfill his dream to travel the roof of the world. Actually, he had a quite big interest in the Asian mountain systems. When I was invited to this event, I was asked to tell a little about my motivation for my research, and particularly on this topic. And it's related to two cities, the cities of Mainz and of Dresden, which have passed the last 30 years, 15 years in each of the two cities. And of course, it's related to my PhD supervisor, Jochen Martens. We can see here with his recording equipment, and Jochen Martens was always sharing his passion for the biodiversity of the Himalayas with his students. So he introduced me in the first research studies on the Sino-Himalayan birds and later on the birds of the Tibetan Plateau, starting with songs, bioacoustics, and then later stepping into genetics. And already during these times, we had good contacts to Dresden, to my predecessor, the former curator of birds, Siegfried Eck. And getting to know the collections of Dresden gave me a great opportunity to do some collection-based research, meanwhile also with our own molecular lab with the clean room facilities that enable us to work on the historical specimens. So yeah, there's a vast opportunity to do some collection-based research here. We have another anniversary, actually, where we have had it a few years ago, celebrating 100 years of biodiversity research on the roof of the world. And at the left-hand side, you can see actually a photograph of a German-Chinese exhibition in December 1913 that started this month. This guy was the leader of the exhibition. He's Walter Stötzner, and he is an ethnologist. He was actually interested in the cultural heritage of Tibet. But the guy here on the left-hand side, he is Hugo Weigold. He was the ornithologist of the team. You might recognize some bird specimens in his right hand. And in a couple of minutes, I will tell you a little more about Hugo Weigold's research. Actually, he brought back big collections of Chinese birds, Germany, dividing them between the Museum of Dresden and the Museum of Berlin, which gave us some work for digitization of his collections in the last couple of years. But we also had the great pleasure to travel China and parts of the roof of the world 100 years after this historical exhibition in June 2013 with our Chinese colleagues. And another big advantage of these historical collections is also that we have these specimens available for genetic analysis meanwhile. And the most interesting part of these collections are possibly the type specimens, which enable us to explicitly tell us how genetic lineages have to be named. This is an example where we genotyped some type specimens of tits from the Weigold collection at almost 100 years old. And last but not least, we had a nice exhibition on 100 years research on the roof of the world a few years ago in our museum in Dresden. What's interesting about Hugo Weigold's studies, actually, we heard that example from Sri Lanka in the last talk, that we have some guys already started biogeography of a certain region of the world. And Hugo Weigold was actually one of the first who really established the theory that Tibet was the center of origin and diversification for many cold-adapted animals. He mainly worked on birds, but he also had some examples from mammals. So he had that ideas of the uplifting Tibet as a diversification center. And he made these observations because he studied the large collections in museums, comparing Palearctic species with the representatives in China, in the Himalayas, also on the Indian subcontinent. As you can see, this is an original drawing from Hugo Weigold, sketching the biogeography of nuthatches, of the Eurasian nuthatches, and its counterparts in Asia, the Indian Peninsula, and the Eastern Himalayas. And it's actually quite well matching the recent phylogeny that we had done for some nuthatch species of this group. Actually, almost a perfect match. So let's start possibly first with the alpine radiation. So we're going right on top of the Tibetan Plateau in the urban landscape and have a look at diversification of alpine passerine birds. We have had a look at this figure actually yesterday in the talk of Raoul Bowie when he was talking about the African arc. Here we see the diversification pattern, the species richness pattern of birds in the world. And you can see the areas of highest species richness in white, and you can easily recognize its three larger mountain systems. It's the Andes, it's the African arc, and it's the Sino Himalayas and its extensions towards the southeast. And what is most interesting is that the white area depicts an area with higher species richness, as well as for very recent species, for young species, as also for ancient, very old species. And if you have a closer look at the Sino Himalayas and compare it to the Tibetan Plateau, we see there's a big difference with high diversity in the mountains of the Himalayas and a very low diversity on the plateau itself. But nevertheless, what you can't see in this picture is that actually many species of the plateau, rather of the old species of the plateau, are very narrow range endemics that occur only there. And the interesting question is, where do they come from? Have they evolved there on the plateau itself? Or did they colonize the plateau from a region, from an adjacent region, being the Palearctic, being the Indian subcontinent, or elsewhere? So here we have a closer look on the Tibetan Plateau with a low precipitation zone in the center, the high elevation plateau, and different mountain forest systems, all biodiversity hotspots in the east, in the south, in the west, and in the southeast. And we had a closer look at one crown clade of the passerines, of the Passeroidea. This is the phylogeny of the birds we were interested in, because the Passeroidea include 5 larger clades of alpine radiations across Eurasia, including the Tibetan Plateau. Here you can have a closer look at those representatives. It's the old world buntings, it's the old world sparrows, the exanthus, the mountain finches and the lice, and the rose finches. So we used the time-calibrated phylogeny. We heard a lot about time calibration in the last talk by Rob Moyle. So we're pretty familiar with that already. And we did some ancestral range estimates for these mountain genera and try to have a look, where do they come from, where their ancestors distributed, and where are their centers of origin and diversification. I'm showing you this in a rather sketched form and then getting to one of the examples in a more detailed form. But actually, all those clades highlighted now have actually diversified in situ, either on the Tibetan Plateau or in the Sino-Himalayas. And also only very recently, during the Pleistocene, 1 or 2 species has dispersed out of that region, either to the Palearctic mountain systems, one example even to the Nearctic, the mountain finches, for example, others to Taiwan. So these are very recent events, these out of Tibet colonizations. But for all these clades, the largest part of the largest time frame of their evolution took place on the plateau or in the Himalayas themselves, and more or less all of them started in the mid to late Miocene. Here's a closer look to one of these out of Tibet radiations, the snow finches actually. And you can see the snow finches more or less separated from their closest relatives, the rock sparrows, at about 12 to 13 million years ago. And the ancestors of the recent snowfinch lineages all evolved in the Tibetan Plateau in the region E with the highest probability for most of the nodes until recent lineages dispersed out of the plateau. And the present distribution actually led to a pattern of symmetry across almost the entire area for these small species. The smaller species more or less diversified on the southern margin of the plateau, whereas the larger species, Montifringilla, they diversified rather on the northern part of the Tibetan Plateau. And if you have a closer look at one of these clades, at the only species that dispersed out of the Tibetan Plateau, Montifringilla nivalis, we can already see that the Asian clade and the European clade is quite diverged actually. And actually, the phylogenetic relationships to that Tibetan species are not pretty well resolved with this multi-locus phylogeny. So in a follow-up project, we were looking on the phylogeography and the population genetics of this larger snowfinch group of the genus Montifringilla. This is a PhD project of one of our students, of Shofi Islam. And he is having a closer look at this distribution. It's the white-winged snowfinch, Montifringilla nivalis, and you can see that it's distributed across the entire Palearctic mountain systems from the northern Tibetan Plateau through the Central Asian mountain systems, the Caucasus, the Near East, until up to the Alps and the Pyrenees and the Cantabrian Mountains. And if you have a look at the mitochondrial lineages, we find just two. We find just two largely separated lineages, Asia and Europe. The Caucasus being very closely related to the Mongolian mountain systems. And down here, we have a data set on SNPs, where we can actually even distinguish between these different mountain systems. We have a clear separation of the Caucasus as a separate lineage. And if we have a closer look in this principal component analysis, we are even able to distinguish between the European mountain system in Spain, the Alps and the Alps. Here you see a time-calibrated phylogeny again. So actually here we can see here is the separation between these species of Montifringilla, the larger snowfinch species, at about 2.5 million years. So that started actually when it got cold. It started with the beginning of the Pleistocene, when these different mountain lineages diverged. And on the right-hand side, you can see a comparison between the different sets of markers, and you can see that the strongest signal actually came from the autosomes. And the autosomes are even capable to distinguish between the Alps and the Cantabrian mountain systems, a signal that you can't find in the mitochondrial DNA or the sex chromosomes. And then we had a look on current and past distributions with the help of Flora Illo and Aidi Niamir, who modeled the past distribution for the late glacial maximum. And there you can see that these larger snowfinch species actually had quite different relic refuge areas. Those areas in green are those with a suitable habitat. And you can see, for example, the Asian populations of the white-winged snowfinch had a quite different refuge area as compared to their relatives on the Tibetan Plateau. And even those are quite different. You have one species with a rather large Pleistocene refuge, whereas the other one has been more or less restricted to the margins of its current range. And if you have a look at Europe, for example, at the European clade, you can see that very probably, this species had more or less vanished from the Alps during the Pleistocene and then recolonized the Alps during the Holocene range expansions from the Iberian Peninsula. So that was a glimpse on the alpine radiations. And if we have a look at the forests surrounding the Tibetan Plateau, we find rather different pattern. Therefore, I'm sketching this in the second part of my talk, having a look at the pattern surrounding the Tibetan Plateau. And this is possibly the most well-known examples of those ring-shaped distributions around the Tibetan Plateau. It's a greenish warbler, which has long been regarded as a so-called ring species. You can easily imagine that the unforested plateau is a large geographic barrier to forest-dwelling birds, and actually many of those have this ring-shaped distribution. The curious thing about this bird in particular is that it has two hybrid zones. It has two contact zones, one in Siberia and one in the Himalayas. And in the northern hybrid zone, you can see here from a genomic study from Al-Qaidi and colleagues that there's almost no gene flow in this northern hybrid zone, whereas in the southern hybrid zone in the Himalayas, you have a very small area of gene flow between a western and an eastern form. You can see it's quite narrow, but this is a typical pattern in the Himalayas that we find areas of secondary contact and at least potential gene flow and hybridization throughout the Himalayas. Here are two examples of these trans-Palearctic circum-Tibetan radiations of the coated above and of the nuthatches below. I've shown you the examples of the nuthatches before, and I will go into detail of the coated examples in the next minutes, because here is actually an interesting area in the Himalayas where you have different phenotypes. It's easy to distinguish the Western and the Eastern phenotype, but in the center, actually, you find a different one. And this is what made us have a closer look at this area, but also because in the Himalayas, you actually find different areas where you have transitions from a Western subspecies to an Eastern subspecies. And these can occur in different large mountain valleys where the Western and the Eastern form have met and now coexist. These can differ a lot. Here are some examples from our own studies on bullfinches with a rather restricted Western form, or from the bush tits with a Western form having a larger distribution range. But actually, it is not well known what happens actually when these birds come in contact. In the coatids, we have a little more information from field studies and from mitochondrial DNA, because we can see that this hybrid zone actually is very small. The blue area here is the hybrid zone. The yellow form here is the Western parent in the Western Himalayas, and the red form is the Eastern parent in the East. And the interesting population were these red-bellied coatids that you find nowhere else than in one particular valley. And you can actually see that according to the mitochondrial DNA, you have only very few at mixed populations. Only in this blue area you have at mixed populations. Then we had, at the next step, we had a look at the microsatellite variation, where you can already see there is larger gene flow between these two areas. The gene flow from that red eastern cluster goes far into the central Nepal populations, even further west into the Himalayas. And what I'll show you next is really absolute fresh data. It's from this week. It's from Shofi's second PhD project. So here we're looking at that particular region using SNPs again, and this is a SNP data set. And you can see that in the PCA, we get actually a quite much better distinction between the parental forms on the one hand, the parental form in the west and in the east, and the hybrids in the center, with some being a separate cluster and some of these clustering with the western form. And those that cluster with the western form are exactly those that have a red belly. The next step would then be comparing these patterns of one hybrid zone with other patterns that occur in the same area with very closely related species. This is the other species pair that occurs in the Himalayas. And the interesting thing is that in the center, we also have a red-bellied form that at the moment is recognized as one subspecies. But it would be interesting to have a look whether there is some, at least some past, some ancient gene flow showing us some ancient hybridizations between that form in the east and that form in the west. So to sum this up a little bit, first on the alpine radiations, if we have read or discussed the phylogenetic relationships and the biogeographic affinities of Tibet, then we often heard about the out of Tibet hypotheses. This is mainly a perspective from mammals, particularly from cold adapted mammals, and you rarely read any papers on out of Himalayas, and not even into Himalayas or into Tibet. This is a rather rare perspective that is just coming up recently, always if we have investigated larger groups of animals. Then you have a perspective that the faunal interchange between the Tibetan Plateau and the adjacent regions is not a one-way street. It's bidirectional, because in these larger groups of birds or of mammals or of plants, for example, you find some clades that diversified in situ in the Tibetan Plateau, and then few taxa dispersed out of Tibet, others diversified in the Himalayas, and only some of them dispersed out of the Himalayas, and so on. But we also find examples of immigrant fauna of animals that diversified elsewhere and then colonized Tibet. And as for the forest radiations, we have these typical ring-shaped distributions in almost all the, in many forest passerines, many more than only the greenish warbler. Here's an example of other warbler species, for example, but you find that in the nuthedges, in the coldhits. But what is very common for these ring-shaped distributions that we have on the southern margin of the Tibetan Plateau, we have east-west disjunctions in the Sino Himalayas. I showed you one example of the coldhits. And on the eastern margin of the Tibetan Plateau, we have north-south disjunctions in China, which are also quite congruent across species pairs, actually. And these are the interesting areas to go to study gene flow and possible speciation processes in secondary contact where these birds meet in the field. And actually, if Hugo Weigelt could have a look at these results today, I guess he would be quite impressed, because many of his predictions actually have been confirmed by recent studies. So there's a lot of partners to thank: students, lab teams, of course, colleagues who donated sample loans for our studies. For example, hello to Rob Moyle again. It's nice to meet you here in this session. You see that the samples from Mongolia that you donated have worked out pretty well. So we keep you informed on the progress of this work. And of course, there's a big thank you to the funding organizations. And of course, again, to the team of Professor Ramakrishnan, Robin, and Vivek for organizing this great event. Thanks for having me with you, and I'm happy to answer any questions.
Thank you for that fascinating talk. We will look for questions. Can people post their questions here?
Actually, at the moment, I can't see the questions box, but if you can, oh, well, I can.
Yeah, I think we,
you may have to start at the bottom.
Sri Lanka. Is there any evidence to indicate the relationships between Himalayan bird species and high elevation Western Ghats species, since there are some floral similarities that have been observed? Yeah, actually, I have to admit that this is a very European perspective I offered, because we have been studying these cold adapted, these montane species, rather from the boreal forests or even from the Alpine plateau. And if you find some relationships between the Himalayas and the Indian subcontinent, for example, the Western Ghats, then these are rather the subtropical taxa at the Himalayan foothills. Yes, there are relationships, also to Southeast Asia, for example, to the Burmese mountains, but also to the Malay Peninsula. You would have to study these subtropical communities, and then you would find very, very different relationships to other parts, to other adjacent regions of the Tibetan Plateau in the south. And this is the Indian subcontinent, and all parts of Southeast Asia, including Eastern China. So is there more?
We have another question. Do you know of biogeographic barriers within
the Tibetan Plateau that impact the speciation? Yeah, actually, within the Tibetan Plateau, there are many, many studies that dealt with Pleistocene processes, so with diversification within species. So those barriers that have been shown within the plateau are almost all of Pleistocene origin. And the usual pattern that you find is that many, many species have been shifted their ranges to the margins of the plateau. As I showed you for one of those snowfinch species, for example. And then some of them recolonized the area after the Pleistocene, and some did not. Some stayed in their, apparently stayed in their relic areas, and we have some endemics that have very, very small ranges. And this is mostly possible, really this is an impact from the glaciation. And let's say from the climate change, because not the entire plateau was glaciated.
Mm-hmm. That's interesting.
Was wondering if you see clades depicting migratory dropouts in the QTP. I'm not so sure what this would mean. It means we had migratory ancestors and then species that stayed there and do not migrate anymore.
That's right, yeah.
Okay. I'm actually not really aware of these things. Actually, many of the species that we studied do not migrate long distances, at least not long distances. Many species on the plateau itself possibly do some seasonal dispersal on the plateau, and many of those forest species actually do elevational migration in the Himalayas, but they're also not long distance migrants. I actually wouldn't have such an example in my own work, and I wouldn't remember another example in the Himalayas.
Okay. We have a couple more questions.
Yeah. What could be cause a difference in degree of hybridization in the Siberian and the Himalayan hybrid zone? I'm not sure about which Siberian hybrid zone we are talking actually, because yeah, if we had a closer look on that coated example, then I've showed only the Himalayan hybrid zone, but there is actually there is a large area of transgression from the Chinese coatids into the Russian coatids. And you can, this is something that you can even see in the phenotype, because the Himalayan and the Chinese coatids, they have a crest. And the Russian coatids, they don't have. And in fact, all those northern and western Palearctic coatids don't have a crest. And there you can see at least one difference, and that means the Siberian Chinese hybrid zone is much larger in extent, because you can see this as a large clinal transgression from a crest to a non-crested form. The patterns of gene flow in that area are not pretty well researched. So we would need a much larger sampling to verify that there's a larger transgression of gene flow from the south to the north. Okay.
We have a question that's moved up because it
Yeah.
was upvoted. So
Yeah,
it's right on
okay.
top.
Which one has moved up?
When you see out of Tibet radiations, do these species retain high altitude adaptations?
Yes, they do. Actually, all these out of Tibet radiations stayed in the mountains. Of course, in most of the cases, they couldn't reach as high as on the Tibetan Plateau simply because the other mountain systems are not that high.
Yeah.
But all of these examples in the Passerodea phylogeny stayed in the mountains. None of these out of Tibet radiations radiated into the lowlands.
Huh.
Okay.
Okay.
What else?
So many.
Is the other current ring species explained by altitude and physical behavior? And interesting work on Himalayan ring species in green warblers, something which has been going on since early 1900s. Is the isolation in current ring species explained by altitude and physical barriers alone, or also foraging behavioral differences recently? This is a very good question. There's a lot of work been done on this issue. Actually, of course, it's not altitude alone, because actually we see that we have a hybrid zone without gene flow in the north, and we have a hybrid zone with gene flow in the south. There have been studies of song divergence, and you can see that song divergence between the western and the eastern form in Siberia in the north is much, much larger than in the south between the Himalayan counterparts. This is one thing, but I think what is much more important is that in the northern hybrid zone, the western and the eastern populations have absolutely divergent migratory pathways. So the western populations migrates west of the Tibetan Plateau, and the eastern populations migrates east of the Tibetan Plateau. And this is possibly the strongest effect separating these populations in the north of the Tibetan Plateau. And this is not as extreme in the south because, as I said, in the Himalayas, these birds migrate altitudinally. So migratory pathways are not as strictly separated as in the north. Okay. What else? I couldn't.
So maybe.
Yeah. Robin, you want to say something?
Yeah, I was just going to say that maybe you could take a few more questions and then let Sampath.
I think there's
jump
one
in
left.
on,
I think there's one
sure.
left. I couldn't see the figure, but Chrysalis was shown to be distributed throughout the arid Tibetan Plateau. What enabled them to occupy the drier areas as well as wetter areas in India and in other countries compared to the other ring species? Well, actually, this is a high elevation species. And also in the Himalayas, it occupies higher elevations, also intruding the semi-open area. So this is not a typical species of humid forests at all, I'd say. Yeah.
All right, so I think we are kind of.
The wall is through with it.
Yeah. If there are other questions, we could always come back.
Great.
Yeah. Thank you. Thank you, Martin.
Thank you so much.
Thank you all for organizing it.
Back to you, Vivek.
Sampath's questions, are we going?
Sure.
Yeah,
Yes.
sure. So
there are quite a few questions about the Sri Lankan Indian distinction.
Nandini, can you
sure.
those, because so many, so I can't remember.
Sure.
I can't kind of initiate where to start and where to.
There's a question about Gondwanan relics present in Sri Lanka today. So could you comment on that?
Yes. Yeah, I saw that, and now I'm kind of lost in terms of where those questions are. But anyway, okay. Talking about Gondwanan relics, in the plant side, I think there's more. They're like, Dipterocarpes, say forest has a fair bit of relics. In the animal side, the cohortes and then the ceratophora, the lizards, they are considered as examples. And then I think like, you know, John, we would tell there are the minor invertebrate phyla that has a fair bit of relics in it. But in birds, some of the relics that, you know, the northern, the relics, the Laurasian relics ended up being not so, those were, you know, like, you know, the taxonomic gaps, phylogenetic gaps actually artificially creating these kind of relics. But the Gondwanan ones are easier in a way because the separation is quite distinct.
Okay.
Yeah.
There's a follow-up question asking about the role of land bridge formation and Pleistocene cycles in exchange of species between India and Sri Lanka.
Yes, the land bridge formation, especially the northwestern part of the island has this the Ramas Bridge, or the series of sandy shores that actually connecting the Indian mainland with the Sri Lanka. That is kind of semi-flooded right now. It's actually like, you know, this like last month one of the islands got kind of submerged, and then about 2 years ago another island got submerged. So slowly I think the sea level rise actually kind of submerging one at a time of the about 20 or so islands. So it is in that cycle where about in the glacial maxima you have like probably a fully like formed bridge or the land bridge, and then in the glacial minima you have basically like a few one or two islands. The real bridge would be the climatic bridge, where climatic differences between the mainland India and Sri Lanka, where the northwestern stretch of Sri Lanka has a kind of arid, dry kind of desert type habitat, which is kind of the same habitat is represented in the Indian side. So in the glacier maxima, where the sea level is substantially low, there is a continuous arid shrublands being formed in 15,000 years or so. So that would trigger some of the lineages to share the two sides, and also these like even still there are small shrub forests in some of these islands in between Sri Lanka and India. So those might play a role in a superficial land bridge. But then you have when you even move into Sri Lanka and into India, there are the reminiscent climatic bridges that you could see along the Indian inland and the Sri Lankan inland, where like we call it icebergs, where definitive small kind of a wet kind of stepping stones that connects the wet zone of South India and the wet zone of Sri Lanka. And in the some of the radiating rivers also into the dry zone also would act as kind of climatic land bridges, even though the course might change a bit, but in a broader geographic, like the broader landscape, those would act as in a way climatic bridges for like facilitating the movement of the wet zone forms into wet zone, and then in between the dry zone forms into dry zone. So it has this, like, the hydrophobic and hydrophilic lineages would react or respond to these changes differently, but it is there. So these changes have been there for at least past 40 million years, where the Sri Lanka is connected and disconnected physically from the Indian mainland.
Okay. There's one more question, actually two more. What are the reasons for rapid radiations in certain species like reptiles and fish in Sri Lanka?
Yeah.
Connected to that, can you comment on the status of marine biogeography?
Okay, yeah, so that's what I was going to say. It's partly to do with the barriers, the strength of the barriers, and how many barriers would act. The ones that are freshwater and the wet specialist would find a whole sequence of barriers from its closest relatives or the landmass where the Western Ghats is, So you have the montane barrier, like lowland barriers, the valleys could be a barrier, and then the salt water, dry climate, like seasonal climate become a barrier, because the wet zone of Sri Lanka, southern wet zone, is a seasonal wet, and then you have the seasonal wet dry areas, and then you have the arid coast, and then the salt water, and then that again represented in the reverse order into the Western Ghats. So that has a lot of barriers, and so the much greater force, pressure for isolation. So the divergence, the potential for divergence, and then the habitat heterogeneity that has been shown in Rockies, in the Andes, and in this case in the Ghats, and in Sri Lanka, where when the habitat heterogeneity is higher, there's great opportunity for divergence, because there's the micro ecosystems started to create. So the western southwestern shore, the face of the mountain range from Western Ghats along the south Sri Lanka, has greater opportunities, because more valleys, more carved out valleys for rivers, and so on. So those lineages that occupy these heterogeneous landscapes, isolated by multiple barriers, would have a greater opportunity for divergence. So it's a mixture of, complex mixture of that. So the certain lineages would capitalize on that and start to radiate, and certain lineages would not capitalize in the same way. That's one thing. The marine forms would have, it started to get complicated, because then you have the larval forms that could move back and forth, and the greater gene flow would you would expect like a slowing down the divergence. But my knowledge on the marine systems are not that good to comment seriously on.
Maybe we'll take one last question. Examples of divergent evolution of fauna from Western Ghats, Far East Asian animals, and what kind of characteristics diverged? And also, are there strong variations in birds of wet and dry zones?
Yes, in a broader sense, Sri Lankan biota is darker in coloration. Partly, broadly you would think that it's the humidity, where, that's been shown in mammals, from people to mammals to the birds. Generally, we have a darker the phenotype found in the as you move south. Typical Glogger's rule and such effects, where latitudinal variation from tropics to north, that effect is there. And on top of that, the humidity gradient is quite substantial. Even in the Western Ghats, Sri Lankan wet zone is wetter than the most part of Western Ghats. So that actually probably causing that, amplifies that the darkness. So that variation is there, and then so with that our forms has generally speaking, in birds, longer beaks, shorter tail, and darker coloration. And in the mountains, some of our mammals started to get bigger, probably for other reasons, the predator-prey dynamics. For example, Sri Lankan leopard is darker in the mountains, bigger and more arboreal, because no tiger. So the other patterns, island effects, started to come into play. So these are not straightforward. There's so many other things come into play. Vocalization, yes. Some of those differences that I was talking about in the birds is actually vocalization. Vocally, they are very different. Again, the same patterns, I think, would go into all these changes.
Yeah, thanks, Sampath. I just want to, Nandini, maybe ask Rob a question, because, having talked about subcontinental birds, Sri Lanka and Southeast Asia, I was wondering if you could add a little bit about what you think about, the evolution of subcontinent, the birds and passerines in the subcontinent are, given your the new findings. Also, what about any fossils? Are there any fossil dates or anything that you've been able to get from this area?
let's see. So first of all, can you hear me?
Yes.
Yes.
Okay, good. The headphones are working again. As far as fossils, I mean, so there's some incredibly rich fossil areas like the Deccan Traps for a lot of groups. I don't think they've found much in terms of modern birds in the Deccan Traps, though, not that I know of. As far as sort of the avifauna, if you think about sort of deep relationships in the Indian region, you know, it's tough because the deep biogeographic history of India probably had some incredible endemicity as it traveled north across the Indian Ocean. It was isolated, at least some. There's some debate about how isolated it was. But after it docked with Asia, that connectivity sort of obscured probably a lot of that endemicity, whether it was obscured via extinction by interaction with the Asian avifauna, or just obscured by diffusion, you know, out into a mixture, is hard to say. India is also somewhat unique in that there's been a lot of change of climate and environment in terms of onset of sort of the monsoon type weather patterns. So a much more seasonal, wet, dry regime, not everywhere, but over large expanses. You also have the influx potentially of avifaunas from Southeast Asia, from temperate Asia, the Himalayas, then even across from Africa as well. So a bit of a crossroads also in terms of the avifauna in the Indian subcontinent region. So it makes it difficult sometimes to decipher deep biogeographic patterns because there's so much mixture going on and changes in bird distributions over time.
Great. Yeah, thank you, Rob, for that, and thanks for staying on, you know, through
Sure.
all the talks as well. Vivek and Uma, maybe we can close with this. I think it's been great having all these 5 days of amazing talks. You know, we got to hear so much about biogeography. It's not easy to get all of these talks in India, and we must thank COVID for this more than anything else. Yeah, so over to you, Uma and Vivek.
Vivek, go ahead if you want to say something.
Okay, no, go ahead, Uma. I'm good.
So yeah, thanks everyone for coming, and it's been really fun, as Robin said, and a whirlwind tour of global, island, all kinds of patterns and studies. And I hope the students have enjoyed all of these talks as much as we have. I think this was kind of an adventure to try this out, and it actually, seems to have worked really well in terms of the tech. There was no glitches or anything like that. And so hopefully this is something we can continue, because we always feel that much disconnected in India in terms of science. Science seems sometimes very Western, and we are far away from it. So I hope that we can continue these interactions, and hopefully that will enrich future collaborations and the growth of knowledge and discovery. So thanks everyone, and thanks to IBS as well for, and Miguel specifically for thinking of Humboldt Day and kind of roping us in and us getting excited about it. Vivek wants to mention something. Yes.
Yeah, so I'd just like to also add that we've had fantastic participation. So we've had more than totally across the 5 days about 1,200 viewers on Zoom, with the first day having almost 450 attendees. And then I didn't realize that, conference fatigue also works on the online forum. So we've also had a steady dropout here. But we've still had quite a few people attending. But also, since we've been live on our Facebook page, that's the NCBS Facebook page, we've had tremendous activity there with almost 10,000 people viewing these talks. And I think that's the first step in inclusivity that we've been talking about. I think this is a great way of, because here now, if you have access to the internet and a cell phone, I guess you can access these talks, and that is really fantastic. So I'd like to thank all the speakers across all the 5 days. It's been a great variety of taxa as well as different regions of the world, and I think it's been really an eye-opener for me especially, and I hope the students have also enjoyed this. Yes, thank you all, and we hope to organize these more often in the future.
Yeah, and thanks Martin and Rob and
Thank
Sampath
you
for the talk today. Yeah,
Thanks
and thanks
for having
Nandini
us.
for moderating.
Thanks, Nandini.
Yes, thank you.
Thank you guys. Yeah.
And thank you attendees. Yeah.
Thanks for your
Thanks,
talks.
attendees. Yeah.
Informative.
Bye, everyone.